Good morning, everyone. Presentation of today is about scientific data management. So within Galapagos, we're looking to see how we can improve the way we are using data and how we can make faster decision making. Before moving into the topic, is it down? Okay, sorry. So for the people who don't know Galapagos, so Galapagos is a biotech company founded in 1999 with a headquarters in Mechelen. As mentioned by Paul, we also have an office here in Basel. I've never been there before, but hopefully this week we will go there. We had quite some changes in the company in the last 3 years. One of our main products, or our first or only commercial product which we had on the market, CDISC, it was decided to sell it or to sell it to another company, other pharma company, to Alpha Sigma. And currently we are in a transition of doing all those marketing authorization holder activities, do the transition to Alpha Sigma. So quite a lot of change for the company. Also impacting a lot of people. Also, the whole commercial organization is moving to Alpha Sigma. I just mentioned that because if you can see on the slides, we are about 600 people in different countries. If you look back a couple of years ago, we were almost close to 2,000 people. So we're coming again a smaller company, and that's also a little bit the strategy from Galapagos to become a more R&D focused company again, where you can see we have 25 programs still in our portfolio, so quite promising there. And also, there's a good reason why we want to go for a scientific data management solution or to work out something like that. Two main therapeutic areas are oncology and immunology. And if you can see here on the left side of the slide, one of our bigger programs is a CAR-T treatment to treat patients with cancer. The unique thing about this is that we do that in a decentralized fashion, where we can treat the people in the hospital in the point of care. And as you can see, that allows Galapagos to treat a patient in 7 days, which is quite faster than if you compare it with a centralized way of treatment, where you also have the shipment towards the central manufacturing site. And I believe there we speak about 20 or 30 days of treatment. And I hope I don't say anything wrong here. The way we can do it is also what you can see here on top, what we call in Galapagos the cocoon. So that's our small manufacturing unit where we can treat or manipulate the blood. And those cocoons are located in these decentralized point of care facilities. In these point of cares, we also have quite some instruments that generate data. The same instruments as we are using in our labs in research. So also here, we could benefit from having a good scientific data management solution to store this data. Of course, here we speak about GXP or GMP processes, so the solution should then also be compliant. So moving towards the topic of scientific data management, so as mentioned before, some time ago, we looked at Galapagos to see how we can improve or make more use of the data. And we looked towards the end-to-end process, starting from how do we generate data and what should we do to have good visualization and analytics. So we divided the end-to-end process in four main blocks. So the first block is everything around. Everything around the data generation, so all the data sources. A second big block is then centralize all the data and store it in a scientific data management solution. The third one is about the data modeling to prepare the data before we can do the visualization and the analytics. So especially the first two blocks here, there we have currently some challenges where we have quite some manual processes still. So all the data generated in the labs by the instruments, we still have people who need to take the data, drag and drop it to a central location, so quite manual, and also prone for error. So, sorry, moving to the next slide. So as mentioned, so quite manual process, not always the most efficient one. It's also time consuming for the people working in the lab, because every time they do an experiment, they need to think about taking the data, put it into a central location. So all the data currently is quite scattered. So we have around 50 instruments, and every person is putting the data somewhere on their personal network drives or a shared drive. Because of this, also we have quite a lack of data integrity, and we don't adhere to the FAIR principles. Also, later on in the process, if data is not organized very well, it's very difficult to have some traceability later on. If we make a decision based on a dashboard, we should, of course, know on which data it was based on. Another challenge for Galapagos is that most of the assays that we do are outsourced to CROs. So around 50 to 80% of our assays are done by CROs. But also then, if that's done by the CRO, of course, at the end, we like to get the data back and store it in a proper way. This is another schema, the same as the 4-box we had before, but in a pyramid, or how do you call it? Where you can see if you want to do some visualization and good analytics, we should have everything below that in a good shape. So starting from all the raw data created by the instruments, storing that data centrally, do some data modeling before we can do good visualization. Also there, if we have this all in good place, we can ensure traceability later on. And again, it's especially about the two lowest levels where we like to improve. Going now step by step to the 4 bigger blocks. If we look to everything about data sources or data generation, so as mentioned before, we have quite some instruments, about 50 in our labs, which all generates data. That data is then synced to the laptop or the computer linked to the instrument. Based on that data, the scientists also do some analytics using FlowJo or Fedra in our case. And also, as mentioned before, all the data coming from CROs, we also like to store that in a more centralized way. So currently, this is all manually. It's all done in our Galapagos network. And we store now the data still already on the central server, I would say, but still it's manually drag and how do you say, drag and paste or drag and copy data from all those different sources to a central server. Second block is about the data ingestion and storage. So from the moment we have the data generated in our first step here, we like to store the raw data, so without doing any manipulations, but store all the raw data files. We like to do some validation checks as well, especially for all the data coming from our CROs, because we have some agreements in case the CRO is providing data, it should be in a certain format. And before we store the data, we like to ensure that the data is in a proper way or in the right format. Then adding some metadata and apply ontologies as well. I will come later back on this one. And then depending on the solutions that we see on the market, you can also do here already some kind of semi-data modeling to ensure that all the assay data or all experiment data is already in a kind of harmonized and structured way. And that then could be then the end for going to the next step is about data modeling. So about data ingestion, as mentioned, currently it's quite manual in Galapagos. We also building our own pipelines and integrations. So there we can, or we should improve to automate it more. We also built some internal developed tool to upload all our CRO data. but also there it's not the idle way. So we want to move away from the in-house developed solutions and pipelines and make more use of what we see already in the industry or what is offered by all the vendors on the market. The same for all the parsers or the integrations. Currently, we also do that all with internal people, but there again, I think many companies are doing the same thing. They have the same challenges, so it would make sense to make use of what is already available in the industry and not doing the whole work again within Galapagos. And then, as mentioned before, also the running of the validation checks before we store the data finally in a central storage. Then some things about data storage. So as mentioned, we want to store all the raw data, and when possible, also do some semi-data modeling already in the second step. And what we then would consider is all the raw data created by our instruments is from the moment they are in this scientific data management solution that we consider it as single source of truth. Because here, that's the only place where we can guarantee that they are safely and stored in a proper way. Then ensure that we adhere to all the FAIR principles, that data cannot be changed. Also enrich the data with additional metadata, where we can say, or to give more meaning to all the experimental data, like when is the data created, for which experiment, with which instrument and things like that. And then also apply ontologies to ensure that all the data that we bring together, that we all speak about the same terminologies. And quite important to also have a scalable solution because we create quite a lot of data. So also there, if you're looking for a scientific data management solution, quite important that we can also scale that on the longer term. The third step is around data modeling and data orchestration. Within Galapagos, currently we are trying to merge all our data in one core data model, so bringing all the assay information in one and the same data format. We do that on a platform which we are currently implementing, and we're almost there to go into production. We call it our qualified data platform, which is built on some different components, so mainly on Snowflake, running in AWS, with some additional tools like dbt for data modeling, Airflow for the orchestration, and then some visualization tools. So goal is here to have this platform qualified so that it also can be used later on for any kind of data flows we like to add, whether it's GXP or non-GXP, so that we have one central data platform here. So as mentioned before, so my background is more in clinical development, and when I joined research, it was quite strange to see that there was no clear structure or standard format for scientific data. Maybe I'm wrong because I'm not so long there in the research area, but if you compare it with clinical development, for example, all clinical trials, they have their CDISC standards, all preclinical, clinical, analytical data, every company worldwide using the same standards, also mandatory in case you want to do a submission to the authorities. I believe here in research or scientific data, there is not such a kind of standard that need to be used by all companies. I found something about Allotrope Foundation, not sure whether it's really the way to go, so it's also more a question to the public here, or to discuss after this presentation to see how other companies are tackling this, because it's still quite strange that every company is developing their own standards. So any feedback here later on, always welcome. And then we have the last part, it's data visualization. I think it's quite clear, so every company can there use their own tooling. We are using FlowJo, Spotfire, but anything, any kind of solution can be used in this step. And to conclude, so what are the next steps for Galapagos? So currently we're still looking around to see what is the best solution or the best way forward for Galapagos to have a kind of scientific data management solution to ensure that we have a clear end-to-end flow for all our scientific data, and to ensure that we have a fast, that we can ensure or shorten the time to insights flow. And the second part, also quite important for us, is to see if we go for a solution, probably we go for a SaaS solution in the cloud, rather than trying to build everything our own again. Because again, every company is in the same boat, I think. And I think it's better to learn from each other rather than trying to reinvent the wheel every time again. And that was my last slide. Good.
Which one do you find the hardest to stand up and to maintain over time?
Is it working again? Yeah. So I think, as mentioned, the second step, where we like to do all the checking of the data, also to add the metadata and apply the ontologies. That's in fact the biggest step, but also the most important one, to ensure that all your data is getting harmonized and standardized. So, yeah.
And have you? One of the other problems that we see sometimes as well is that ontology will change over time. So not only do you need to clean your data, check, do the validation checks, fit it to the ontology, but then we say, we have another question that we need to answer, and we need a new part of the data model. Then you have to go back through and re-enrich or re-ingest the data. So is that another consideration that you guys have thought about?
Yeah, and that's what we all consider as part of that second step.
Right. Yeah.
Where we also try to rely on what vendors are offering there. Yeah.
Because it's not easy.
No, and it's also a big effort. So.
Because then you end up with that version. Which version did we make the decision on, and which version is live, and things like that. So.
So also they're quite important indeed. If we have that scientific data management solution, that we also keep all the versioning and the history of all the changes. So yeah.
Just gets bigger and bigger and bigger. Any other questions?
One point.
Yes.
Devendra from Thermo Fisher. Quick question. Can you elaborate a bit more on what your concerns are with allotrope? What's your hesitation or?
Yeah, I don't think we have really concerns on the allotrope foundation. It's more that we don't see any standard that should be followed by all companies. So I think there are different initiatives. This one was one example, but I don't believe that every company is using this one as their standard. So I think it makes sense that we have one standard for all scientific data, the same as I referred to for clinical data, where we have the CDISC, which is used commonly worldwide. So, yeah. But I'm not so familiar with this standard yet.
Yeah, because you've got others, you guys know, right? For sure, it's animal. You've got all sorts of other formats.
So you're using other standards or other guidance. Yeah.
Christophe Puzna, Science Co. Just some question relating the sources. How many kind of instrument you have to manage with different brand, different software, different instruments technology? Because we know there are a lot of things to do, but I don't know the number of challenge you have depending the nature of the measurement you do.
So if I understand correctly, so we have a kind of 50 different instruments that we are having in our labs. And as mentioned, also we like to store all the analyze the data coming out of Fedora and FlowJo, for example, which is also, again, another format. And then also all the data, all the assay data that we receive from our CROs. I think it's around 3 or 4 CROs we're working with. We always try to ask them to provide the data in the same format, so that we are already standardized. Sorry, but in the end, yeah, I think we speak about 50 or 60 different formats that we need to process.
Thank you.
Yeah.
Was your question also about the type of instrument? So you have benchtop, like pH meters, balances. Does that come under that purview, or is it more of the advanced analytical process instrumentation, or is it everything?
I assume it's everything, but I don't know the exact names of all the instruments that we have. But,
Yeah. Can guess it's a lot.
yeah.
Yeah. Okay.
I had a question.
Any?
Yeah.
Oh.
One
Yes,
here.
you have the mic. Look at that. Yeah, you're in charge.
Hi, Zishan. Just was wondering, because you write here, move away from in-house built solutions, so everybody has different reasons for that.
Yeah.
But what are your personal or business reasons for moving away from in-house built solutions, and what do you kind of look for in not in-house built solutions?
Yeah. I think the main thing that we see that it's also a big effort to do that in-house, and you need the people. But I believe the most instruments that we are using, they're also used by other companies, and they also need to do the same kind of activities to bring the data to a central platform. So there we also say, okay, why are we building our own pipelines or our own parsers to move the data? Because I believe there are many vendors already who are offering standard parsers that can do that activity.
Thank
So
you.
it's just a matter of using best practices or what's available. No.
Another question? Yep.
So yeah, my name is Harpreet. I'm from SiEE. So you mentioned that you have agreements with the CROs to have some sort of format that you finalize for them to deliver the data.
Yeah.
Do they add the metadata before they deliver the data to you? Is there any kind of ontologies that you have standardized for them that they have to use when they submit that data to you?
It's a question I cannot answer, so I don't know the exact answer, but I believe we only ask the raw data, and we will add the metadata and ontologies later, because we also need to harmonize it with our own internal data. So it's always, we can ask probably quite already to provide it in the standard way, but anyway, we need to do some work also at our side to also bring our assay data together and to add metadata and ontologies.
Thank you.
Yeah.
Excellent. Roman.
Does that? Okay, that works. The ideas you presented, you said you want to aim at accelerating decision making and more generally research processes. And I'm curious, how do you measure these types of outcomes, and how much have you already achieved in that acceleration?
Yeah, so I think the goal of the presentation indeed was more that we see that there is a need, so we are not there yet. Currently, the challenge is that we have quite some manual steps still to do, and we're now looking to implement a scientific data management solution to do it faster and to do it more efficient. But we're not there yet, but hopefully the sooner the better.
Do you have any plans on measuring this acceleration, or do you have any ideas at least? Because I think this is really difficult, and that's why I'm curious what your thoughts are on doing this, to really also test whether what you're investing into these ideas actually translate to those outcomes you want to achieve.
I think so far we didn't have any matrix or KPIs in mind, but it's certainly something indeed to work on. Thanks.
I think that's a really good one for the break, because I think that's something that a lot of people think about, and we say we're going to impact the business, we say we're going to impact science, and we're going to have efficiency, but how do we actually measure that is hard. Yeah, it's hard.
And that's also always the discussions we have also within our company to see what is the business value or the business case to work on this, and it's indeed very difficult to measure really what is the benefit. So, very good question.
If one more question at the back there, and then we'll...
Thank you so much. Quick question. You were talking about data and data modeling a lot. How do you capture data in the lab, actually?
Sorry, how do you?
How do you capture data? Do you have a notebook?
Yes, all data is going to our ELN. We have 2 ELNs at the moment, depending on the kind of information, as we have one for chemical and one for biological. But of course, it's the idea to bring that into one solution. But currently, we have two ELNs available. Yeah.
Thank you.
Yeah.
Okay, so Kris, thank you very much.
Okay, thank you.
Cheers. Thank you.